Everyone, thank you so much for joining us today. We appreciate you taking out the time to participate in this webinar. I'd like to welcome you all to our first ever 2017 collaborative webinar series. My name is April Latson. I am the Community-Based Organizations Outreach Coordinator for the SiNERGe project, and I represent the William E. Proudford Sickle Cell Fund and will be your moderator. With these presentations, we are hoping to provide the most current information on a variety of topics of particular interest to the sickle cell community. These webinars are open to all interested parties, and we welcome your input regarding any topics of interest to you in the future. At the end of the presentation, I will be reading questions from the audience. Please feel free to post your questions in the group chat box on your webinar page, or if you are joining us by phone, please feel free to email me your questions at alatson@wepsicklecell.org. That's alatson@wepsicklecell.org. alatson@wepsicklecell.org. Today, we will explore information on bone marrow transplant and gene therapy. Our presenters today on bone marrow transplants include Dr. Jennifer Krajewski and Dr. John Horan. And presenting on gene therapy, we will have Dr. Michelle Lee and Dr. Erica Ezrick. Okay. So one disclosure, the views expressed by presenters are their own and do not necessarily reflect the views or opinions of the William E. Proudford Sickle Cell Fund, the SiNERGe project, or associated parties. This webinar is for informational purposes only. Individuals seeking medical advice should always see a qualified health professional. Our presenters will share professional insights, accounts, and experiences regarding bone marrow transplantation, clinical trials, and gene therapy. Our first speaker will be Dr. Jenny Krajewski. Dr. Krajewski has served the Pediatric Blood and Bone Marrow Transplantation Program at Hackensack University Medical Center since 2010. Her primary interest is in the use of allogenic transplantation for sickle cell anemia. Board certified in both general pediatrics and pediatric hematology oncology, Dr. Krajewski conducts research and publishes in her field. Dr. Krajewski earned her medical degree at Finch University of Health Sciences, the Chicago School of Medicine. She completed her general pediatric residency at the Children's National Medical Center in Washington, D.C. Along with STAR, her professional affiliations include Alpha Omega Alpha, the American Society of Blood and Marrow Transplantation, and the Pediatric Blood and Marrow Consortium. Our second speaker on bone marrow transplantation and clinical trials will be Dr. Horan. Dr. Horan is an associate professor of pediatrics in the Emory University School of Medicine and an attending physician in the blood and bone marrow transplant program at Children's Healthcare of Atlanta. He is the medical director of STAR, Sickle Transplant Alliance for Research, an organization dedicated to advancing the cure of sickle cell disease through blood and marrow transplantation. Speaking to us regarding gene therapy is Dr. Michelle Lee. Dr. Lee is an instructor at Harvard Medical School in the Department of Pediatric Oncology at Dana-Farber Cancer Institute and the Division of Hematology Oncology at Boston Children's Hospital. She is a clinical expert within the Stem Cell Transplant Disease Center. Dr. Lee attended Spelman College, where she earned a Bachelor's of Science in Biology. She earned her MD and PhD degrees at Harvard Medical School and Harvard University. She trained in general pediatrics at the Children's Hospital of Philadelphia, in the pediatric hematology oncology at DFCI BCH. Dr. Lee's clinical interest is allogenic stem cell transplantation for patients with non-malignant hematologic diseases, including sickle cell disease and bone marrow failure. Her clinical research interests are in improving donor selection for patients with poor alternative donor options. We will also have Dr. Erica Ezrick. Dr. Ezrick is an instructor at Harvard Medical School in the Division of Hematology at Boston Children's Hospital and the Department of Pediatric Oncology at Dana-Farber Cancer Institute. She is the clinical leader of the thalassemia program at BCH. Dr. Ezrick earned her BA in biology at Dartmouth College and her MD degree at Harvard Medical School. She trained in general pediatrics at the Boston Combined Residency Program and in pediatric hematology oncology at DFCI BCH. Dr. Ezrick's clinical interest is in caring for children with hemoglobinopathies, including sickle cell disease and thalassemia. Her clinical research interest is in the development of clinical trials for gene therapy and other novel therapeutic options for hemoglobinopathy patients. I'm now going to turn the presentation over to Dr. Krajewski so she can talk to us about stem cell transplant, a cure for sickle cell disease. Dr. Krajewski?
Good morning, everyone. I have nothing to disclose. I'm going to talk about 3 objectives. The first is going to be just to review the basics of stem cell transplantation. The second is going to be to review the history of transplant as a cure for sickle cell disease. And then the third will be to review the latest studies on transplant for sickle cell disease. So what is a transplant? The definition is simply giving a patient someone else's stem cells as a cure for a disease. When we talk about transplant, there's a couple different things that we need to discuss. First, our donor options. This really includes either a related donor or an unrelated donor out in the world. Ooh. Sorry, guys. Seems to be advancing.
Liverpool history, the matchup.
The second thing that's important in transplant is HLA matching. So these are the genetic tests that determine how compatible a patient and a donor is. We test 6 or 10 alleles, and the patient and the donor can either be fully matched, mismatched. I don't know why it's advancing, I'm sorry. Okay. You can either be fully matched, mismatched, or what's called haploidentical, which means matching by half. And then there are 3 options for your stem cells: bone marrow, which is the most common, peripheral blood stem cells, and umbilical cord blood. There are 3 primary complications of stem cell transplant, no matter what you're doing the transplant for. The first is something called graft versus host disease, or GVHD. And that's where the donor cells recognize that the patient is not where they usually live, and they attack the patient's body. The second is something called graft rejection. That's when the patient's immune system prevents the transplant from growing. And then the last is infection, and this is due to the medicines that are needed in order to perform transplant and prevent the GVHD and graft rejection. So to prepare the patient, we need to do a couple different things. The first is you need conditioning in order to decrease the patient's immune system and prevent rejection. We use chemotherapy and various antibody treatments targeted against the immune system to do this. And these can either be myeloablative, basically meaning you're completely wiping out the patient's immune system, or using reduced intensity, where you're giving enough therapy to decrease their immune system, but you're using less toxic things. And then the important thing to remember is that the patient needs immunosuppression for at least a few months after transplant in order to prevent that graft versus host disease I mentioned. I just want to mention a few important terms that I'll discuss when we talk about the different studies. The first is overall survival, or OS. This is the chance of surviving the transplant. Second is disease-free survival. This is the chance of surviving the transplant without the disease that you're doing the transplant for. And then the last is EFS. This is the chance of surviving the transplant without the disease or other significant complications. So now we'll talk about bone marrow transplant specifically for sickle cell. The first one was done in the 1980s in a patient that had leukemia, and they found that not only did they cure the leukemia, but they also cured the sickle cell disease. So Dr. Walters did the first study looking at this. It was a national study that included 50 pediatric patients, all with severe sickle cell disease, meaning these were patients that had had stroke, recurrent pain crises, or recurrent acute chest. They all had fully matched sibling donors. They used that very intensive myeloablative conditioning that I talked about, and the overall survival was 94%, which was very good, meaning the majority of the patients survived the transplant, and the event-free survival was 84%. So not all of the patients that survived had cure of their disease. More recently, earlier this year in 2017, Gluckman looked at 1,000 matched sibling transplants done for sickle cell disease between 1986 and 2013. He found that 87% of the patients received that myeloablative conditioning, 84% received bone marrow. The 5-year overall survival was similar to that initial study at 93%, but the event-free survival was much, much better at 92%, meaning that almost everyone that survived the transplant was cured of their disease. And one of the key things was that only 23 patients experienced that graft failure. So reduced intensity conditioning has been a newer thing in the field of sickle cell disease, with the goal being that if we can reduce the intensity of the conditioning, the side effects when you're going through the therapy will be less, and also your long-term side effects will be less. And one of the biggest things is that reduced intensity conditioning hopes to preserve the fertility of our patients. We know patients that undergo myeloablative conditioning likely will not be able to have children in the future, but there is early data that reduced intensity conditioning will allow patients to have children in the future. There was a recently published study back in 2015 that looked at the use of reduced intensity conditioning for 52 patients who underwent matched sibling transplant for hemoglobinopathies. Most of these patients, 43 of them, had sickle cell disease. The overall survival was 93%, and the disease-free survival was 91%. So very successful with minimal issues of graft rejection. The 3 deaths were from graft versus host disease related issues. So unfortunately, only about 14% of our sickle cell patients have a sibling donor that doesn't also have sickle cell disease. So that leaves a big proportion of our patients that don't have sibling transplant as an option. Because of this, there was a study that was conducted in the mid-2010s that looked at unrelated donors for children with severe sickle cell disease using this reduced intensity regimen. It included 30 patients ages 4 to 19. The indications for their severe sickle cell disease were stroke in 12 patients. 2 patients had abnormal transcranial Dopplers, which we know is a risk factor for stroke. 12 patients had recurrent pain crises, and 4 patients had recurrent acute chest. All of these patients received 8 out of 8 fully matched unrelated donors. Unfortunately, the results were not as encouraging as we had hoped. The 1 and 2 year event-free survival rates were only 76 and 69%. And the overall survival rates were only 86% and 79%. So significantly different than the results seen with sibling matches. The reason for these results relates to the GVHD. So acute in order to give more patients the option for unrelated transplant. Sorry, my computer just died for a second. Okay, so the next question is, can we use non-myeloablative conditioning, or sorry, can we perform transplant for adult sickle cell patients? The thought initially was that adult patients would be too sick to be able to tolerate transplant, but we knew that we were missing a big subset of patients that could potentially benefit from this cure. So a group back in 2000, in the late 2000s, looked at using non-myeloablative conditioning, so extremely low intensity conditioning, and matched sibling donors for adult sickle cell patients. So currently, there's been 41 patients that have been enrolled to date. The age range is very large, as you can see. It's ages 17 to 65. The incredible things have been, there have been no treatment-related deaths. There's actually been no acute or chronic graft versus host disease. 20 of the 31 patients that are eligible to be off immunosuppression have been taken off their immunosuppression. And there's actually been 7 children reported from 4 patients to date. So far, in adult patients with sibling donors, it does seem like transplant is a viable option. Looking into this further, there was a pilot study that was done looking at the safety of transplant in patients ages 15 to 40 with severe sickle cell disease. 23 patients were enrolled. This transplant protocol actually used myeloablative conditioning, so more intensive conditioning. The donors consisted of 17 matched sibling and 5 matched unrelated. And the overall survival and event-free survival were both 90%, which was very good for this high-risk population. So because of this, there's actually now a study called STRIDE-2 that is open. It's a multi-center study across the country that is looking at determining the safety and efficacy of transplant versus the standard of care in adolescents and young adults with severe sickle cell disease. Basically, if you are eligible for this study and you have a donor, you will go to transplant, and if you don't, then you will continue to receive the standard of care supportive treatment that you're currently receiving. And the last question I wanted to discuss is just, should we be performing BMT in patients with sickle cell disease, and is there an advantage in doing so? Bhatia et al. Back in 2015 measured the quality of life before and after stem cell transplant in patients who underwent reduced intensity bone marrow transplant. There were 17 patients and 23 caregivers enrolled because these were pediatric transplants. In the patient-reported analysis, there were significant improvements in the overall health-related quality of life at 1 year post-transplant, and the same trend was seen in parent-reported overall health-related quality of life. So it does seem to improve the quality of life if you undergo transplant. One of the other key things is, does this improve the healthcare utilization for our patients receiving stem cell transplant for sickle cell disease. Arnold et al. Back in 2015 looked at 26 stem cell transplant patients and 48 control subjects from 2002 to 2011. The healthcare utilization cost of transplant was determined over 3 periods. There was pre-transplant, during transplant, and post-transplant. And the post-transplant stem cell transplant healthcare utilization was decreased when compared with both the pre-transplant costs and with the cost for the control subjects. And I think the best way this is illustrated is by looking at this graph. The control subjects are here in red, and as you can see, there is a significant improvement between, or significant difference between the control subjects in every area. So their inpatient length of stay, the number of inpatient visits, and the number of the cost of outpatient visits, comparing control subjects with those who had undergone transplant. So it does seem that both from a quality of life and from a cost perspective, transplant is beneficial. That's all I'm going to talk about. We'll take questions later on in the talk. Thank you so much for participating in our webinar today.
Yes, I just wanted to let you know a little bit more about STAR. So next slide. The Sickle Transplant Alliance for Research is something that came together. We came together as a group informally a few years ago. Originally, it was mostly pediatric bone marrow transplant physicians. Then we added a number of sickle hematology doctors. And in December 2015, we became a 501c3 charitable organization. We have an executive director, Ruth Dinnerman, who I think is on the phone. We have a board that's actually increasingly comprised of non-medical people, people from the sickle cell community and others. And we've grown now to actually up to 18 centers, and that includes 2 in Canada, 1 in Calgary, and 1 in Toronto. And our mission is to enhance the lives of children suffering from sickle cell disease through blood and marrow transplantation research to create better transplant approaches, ones that safely achieve cure for the greatest number. And our focus on that safety is really, Dr. Jenny talked about graft versus host disease and fertility, and 2 of our goals is really to come to as close as possible to eliminating that risk of graft versus host disease and to do transplants without affecting the fertility of the patients with sickle cell disease. Next slide, please. Okay. So that's just a map of the centers, and then next slide. Here's just photos of number of patients who've had transplants at STAR centers. A couple of kids on there from one from Atlanta, several from Columbia, one from Nationwide, UNC, Calgary. So these are all children that have successfully gone through transplantation. I think most of them, if not all of them, have received match-related donor transplants. Next slide. So just to introduce you briefly to the 2 trials that we've opened. So the first is called Abatacept for Preventing Graft versus Host Disease. And the doctors that are leading that are Monica Bhatia from Columbia University and Dr. Sonali Chowdhury, who is from Northwestern University, Lurie Children's Hospital, Chicago. And this trial is open to, so we know from work in sickle cell disease, bone marrow transplant for sickle cell disease, as well as bone marrow transplant for leukemia, which is far more common, that we have a pretty good idea of who's at most risk for getting graft versus host disease. And so we can tailor our clinical trials to those patients. And this is an example of that. So we're using Abatacept, which is an immunosuppressive medication. It's been around for about 10 years actually a little bit more. Originally was developed for use in the treatment of adults and then children with rheumatoid arthritis. And then several years ago, we not being STAR, but several of us who are part of STAR, but through a different a group, started investigating this medicine for the prevention of graft versus host disease in adults and children who are getting transplants for leukemia. And so we just completing a trial of nearly 200 patients in that setting, and the results are very, very promising. And based on those preliminary results, we've started to open up trials to other groups of patients, one of those being children with sickle cell disease. So this trial is open to children who are severely affected by their sickle cell disease. They also have to be at high risk for graft versus host disease, so they have to be either getting an unrelated donor transplant, and Dr. Jenny showed before that those patients have a high risk for graft versus host disease, or they need to be an adolescent patient or have an adolescent donor. So those are all things that predispose to graft versus host disease. And we opened this trial in the fall. We took a break over the winter respiratory viral season, so that we weren't taking patients through transplants and weakening their immune system, so they would potentially putting them at risk of bad cases of flu. So we now restarted. We did four patients in the fall who are all now doing well, and we've just started treatment on 2 more, and we have a number of other patients lined up for the spring, and we hope to complete this trial by the end of the year, by the end of next fall, and it's going to be 20 patients altogether. And hopefully from that we'll build and even go on to, if the results are encouraging, we'll go on to do a larger trial. And then the next slide, please. This is a different sort of trial, but kind of again build on that, the importance of understanding the risk for graft versus host disease. So we increasingly have parents that once they learn that their child has sickle cell disease, they begin to explore bone marrow transplants. And so centers are starting to perform transplants for kids that are even 2-3 years old, who really haven't developed a lot of problems from sickle cell disease. And this kind of takes that, puts it into a clinical trial, and the idea is opening it to kids who have not really developed severe problems yet, but restricting it to kids that we know are very, very low risk for graft versus host disease. And so that's kids under the age who are not adolescents and have a match-related brother or sister who's also not an adolescent. And we know that the risk of serious graft versus host disease in that setting is very, very, very low. The trial is being done with a reduced intensity conditioning regimen to preserve fertility. And that trial should be, it's open, and we should be enrolling patients in the next few months. The trial is being led by Dr. Kazow at the University of North Carolina and Emily Meyer, who's in Indiana. And this is the last slide, please. It's just if you'd like to learn more about STAR, this is from Ruth Dinnerman, our executive director. Again, the website address, and if you'd like to receive updates, a link down at the bottom. So that's it. Thank you. And next up is Erica and Michelle, who will be talking about gene therapy.
My name is Michelle Lee, and I'm here with Erica Ezrick, and we're glad to have the opportunity to talk today about gene therapy in sickle cell disease, a new potential for cure. Our disclosures are described here. So curing sickle cell disease means preventing the sickling of red blood cells. Sickling red blood cells cause hemolysis and vaso-occlusion that lead to pain and organ damage in sickle cell disease. If we prevent sickling of red blood cells in patients with sickle cell disease, they can be cured. You've already heard this morning about how bone marrow transplant can be a cure for sickle cell disease, And you've also heard about how bone marrow transplant options are determined by donor availability. So what I mean is that when we have a person with sickle cell disease who has a healthy HLA-matched sibling, that person can consider undergoing bone marrow transplant from their HLA-matched sibling. But you've also heard that most patients with sickle cell disease do not have an HLA-matched sibling. And for those persons, they can consider stem cell transplant from an alternative donor. The important issues are that a donor source has to be identified, and that donor source could be an unrelated volunteer donor, a haploidentical related donor, or a cord blood unit. And up until recently, those were the only options available to people who did not have a healthy HLA-matched sibling. More recently, though, gene therapy is a new way to prevent sickling of red blood cells and to possibly cure people who have sickle cell disease.
This is a slide depicting, just with pictures, the general concept of gene therapy. So if you look in the bottom left of the picture of a sick patient or a patient with sickle cell disease, moving upward, the HSCs, or hematopoietic stem cells, are removed from that patient via bone marrow harvest. And then those cells are transduced with what is called a viral vector. That is a virus that's inactivated and harnessed to carry the genetic material that we want to insert to fix the disease. Those transduced cells are then transplanted back into the same individual, so it's his or her own blood cells, and hopefully resulting in a cure, or at least an improvement. At the bottom, it also shows that that patient has to undergo conditioning similar to an allogeneic transplant before receiving his or her own blood cells back.
So Dr. Ezrick and I have two parts of our discussion today. The first is comparisons of gene therapy and alternative donor stem cell transplant, and the second describes in a bit more detail how gene therapy works. So first we'll talk about comparing gene therapy and alternative donor transplant. We'll consider conditioning regimens, risks of infection, graft versus host disease, graft rejection, and leukemia. And lastly, I'll talk about eligibility for undergoing gene therapy. So comparing conditioning regimens. So conditioning regimens are the medications that are given to patients prior to receiving the infusion of stem cells. And the purpose of the conditioning regimen is to make room in the bone marrow and the immune system for the patient's body to accept the infused cells. Gene therapy uses full intensity conditioning to allow engraftment, or to allow those new cells to grow in the patient. An alternative donor transplant most often uses reduced intensity to allow those new cells to grow in the patient. With gene therapy and full conditioning, the risk of organ damage is more than an alternative donor transplant, and the effects on fertility are also likely to be more than alternative donor transplant. In these slides, I've used red to indicate higher risk issues and green to indicate lower risk issues. And with respect to conditioning regimens, alternative donor transplant has lower risk. With respect to risk of infection, though, gene therapy has less risk. So after conditioning is given, the patient's blood counts fall, and the patient is at more risk of infection. And there are two different ways that we think about infection. One is neutrophil engraftment. So neutrophils are the cells that fight bacterial infections, and those cells are low after conditioning, and then they recover. Neutrophils are important for fighting bacterial infections, and in gene therapy and alternative donor transplant, neutrophil engraftment, as far as we understand right now, occurs at about the same period of time. However, another way of looking at risk of infection is how fast the immune system recovers. This is a different part of the immune system that mainly is involved with fighting viruses. So after gene therapy, because gene therapy involves using the patient's own cells, immune recovery is fast, different from alternative donor transplant, where immune recovery is slow. So after gene therapy, patients may have less or fewer infections than after alternative donor transplant, where they may have more infections. We've talked a little bit about graft versus host disease, and the risk of graft versus host disease is significantly different between gene therapy and alternative donor transplant. Graft versus host disease is when the new donor immune system attacks the patient's body. So in gene therapy, because the cells that are being infused into the patient are the patient's own cells, there's no risk of graft versus host disease. On the other hand, in alternative donor transplant, there may be a significant risk of graft versus host disease. That means that the donor immune cells can attack the patient, and the things required in alternative donor transplant to minimize that risk are prevention medicines, but patients are also at risk if they develop graft versus host disease of requiring treatment. So this is just to say again that in gene therapy, there's actually no risk of graft versus host disease versus a possible significant risk in alternative donor transplant. We also talked earlier about graft rejection. So graft rejection describes whether or not the cells that are infused actually continue to persist inside the person who received them. With respect to gene therapy and alternative donor transplant, this has remained a challenge. So in gene therapy, as this therapy is actually rather new, the incidence of graft rejection is not yet entirely known. And we expect over a hopefully short period of time to have more information about this. On the other hand, in alternative donor transplant, graft rejection has sometimes been a significant problem, but it's a problem that the field is working on. So the issue of graft rejection and whether or not the transplant takes in both of these circumstances may have particular challenges. And then finally, I wanted to compare risk of leukemia. So it's very challenging when we're talking about transplant or gene therapy to bring up the concept that someone can get a treatment for one complicated disease like sickle cell disease, and that it may put them at risk of another complicated disease like leukemia. The risk of developing leukemia in either case is small, but it still exists, and that's why it's important for us to discuss. There are different ways that patients can develop leukemia after gene therapy or alternative donor transplant. After gene therapy, the full intensity conditioning gives a small risk of patients developing leukemia in the future. And also, because genetic material has been inserted into the patient's own stem cells, there's a theoretical risk that that can cause leukemia. That's compared to alternative donor transplant, where the reduced intensity conditioning is associated with a very small risk of leukemia. So there's a difference here, but the risk of leukemia from either procedure is likely to be low. Finally, I just wanted to talk a little bit about eligibility for receiving gene therapy or alternative donor transplant. So for patients with more severe sickle cell disease who have a sibling donor, transplant is actually considered a standard of care. However, for patients with more severe sickle cell disease who do not have a sibling donor, either alternative donor transplant or gene therapy would likely be performed on a clinical trial. There is a lot of information available about what a clinical trial is and what it means to participate in one. But the reason that clinical trials are being used in these circumstances are that so that we can both treat people and also learn how to treat future people better.
I'm just going to shift gears here a little bit and talk about some specifics of the strategies for gene therapy for sickle cell disease. So this figure shows an example of a red blood cell, and in the red blood cell is a picture of the beta globin gene complex, including the green square, which represents the mutated sickle globin. Here are two general strategies for gene therapy. So gene addition is where entirely new genetic material is added to the cell. An example of that, I'm going to show a few currently open gene therapy sickle cell trials. An example of that is a trial that's currently open, sponsored by the company Bluebird Bio and open at a number of sites around the country, in which a vector, which is the virus that which is the genetic material that's inside the virus that I described, contains a modified version of normal beta globin. It's modified in a way that makes it anti-sickling or helps prevent the sickling process. So the picture to the right simply depicts the details of the viral of the vector that's inserted in this trial. This trial includes conditioning with busulfan only, and the first patient that has achieved a follow-up of about 15 months was reported recently in the New England Journal of Medicine. This figure is from that report, and I'll just walk through it. So all this data is about the types of hemoglobin that the single patient in that paper achieved. So across the bottom, you have the number of months since gene therapy. And across the vertical axis, you have the amount of the different types of hemoglobin. So looking first at the blue line, which is hemoglobin A or normal hemoglobin, it was high at first simply because that patient was receiving blood transfusions, and then declined as that patient required fewer and fewer blood transfusions over the next months. The green line represents sickle hemoglobin, which started very low because the patient was being transfused, and then increased over time to an amount of 49%, which is far less than that patient with hemoglobin SS would normally have without transfusions, which would normally be up around 80 to 90%. And the black line represents the gene therapy product. So the hemoglobin that was derived from the genetic material that was added, which also achieved approximately half of the hemoglobin. So in other words, the genetic makeup of this patient is now far more similar to a patient with sickle cell trait, and thus asymptomatic compared to hemoglobin SS. And finally, to point out that the total hemoglobin achieved was 11.8, which is, as you know, far higher than a typical hemoglobin level for a patient with hemoglobin SS. Back to the list of open trials, there's an open trial at UCLA, which, similar to the first one, adds a modified beta globin gene that's also an anti-sickling globin, similarly uses a busulfan conditioning regimen and has not reported any results yet. And a trial in Cincinnati that's also a gene addition approach in which a gamma globin gene, which is the one of the genes involved in fetal hemoglobin, and is added. And sorry. This also has no is open but with no results reported yet. Oh wait, sorry, there we go. So returning to the strategy slide, the other main strategy being considered for gene therapy is, instead of adding entirely new genetic information on top of the sickle gene, instead inducing the expression of fetal hemoglobin. high levels of fetal hemoglobin make sickle cell disease less severe. This has been known for decades. And so, for example, in this figure, which is from a natural history study of sickle cell disease by Platt et al. In the 1990s, shows that over time, patients who naturally make a high level of fetal hemoglobin survive longer than patients who naturally make a low level of fetal hemoglobin. So for decades, increasing fetal hemoglobin has been a core strategy in the treatment of sickle cell disease, including treatments such as hydroxyurea. And in many studies over the decades, it has been clearly shown that increasing levels of fetal hemoglobin on average decreases the problems in sickle cell disease. BCL11A is a protein that was recently discovered to be an important regulator of fetal hemoglobin levels. So this is a picture from a preclinical study performed on mice. So on the left, you have a control or a normal mouse model. That's the red blood cells of that mouse. In the middle, you have a mouse model of sickle cell disease. So you see the sickled cells, the target cells, and far fewer blood cells because that mouse basically has sickle cell disease. On the right, you have that same sickle cell disease mouse model where the protein I described, BCL11A, has been knocked out. And you see that the red blood cells are transformed to far more normal in appearance. So the conclusion is that the knockout or the knockdown or inactivation of BCL11A increases fetal hemoglobin levels and therefore corrects the defects in sickle cell disease. So this would be an appealing strategy for treatment in humans. That strategy forms the basis for an upcoming gene therapy trial that will be opening at our center, which is an example of the other main strategy for autologous treatments for sickle cell disease, increasing fetal hemoglobin. So at the top, you see that the goal is to decrease BCL11A and therefore increase fetal hemoglobin. And all the way down at the bottom is just depicted a kind of a simplified version of the viral vector, of the vector that would be included. And in the middle is what's called a short hairpin loop that is a strategy for knocking down or inactivating BCL11A. So again, to just go through the picture in the middle, the patient treatment across the top, the patient has bone marrow collected to collect those hematopoietic stem cells, undergoes conditioning with busulfan, and then has the cells infused again. And at the bottom, at the same time, those blood stem cells or hematopoietic stem cells that were collected are transduced with the vector that knocks down BCL11A and increases fetal hemoglobin, and then is ready for infusion back into the patient, him or herself.